There is something very difficult to believe about cancer. In 1975, only about 57% of children diagnosed with acute lymphoblastic leukemia, or AL, survived at least 5 years after diagnosis. Today, that number has risen to around 92%. But now look at glioblastoma, a highly aggressive form of brain cancer. For this disease, the median survival time for patients is still only around 15 months, and the 5-year survival rate remains below 5%. Both diseases are called cancer, yet one has seen remarkable improvements in its outlook, while the other remains extremely difficult to treat. Why? And if humans already have surgery, chemotherapy, radiation therapy, targeted drugs, and immunotherapy, why haven't we found a way to cure cancer for everyone? Before answering that question, there is something we need to clarify. Saying that humans cannot cure cancer is actually not entirely accurate. We have achieved major successes with many types of cancer. Differentiated thyroid cancer is one example. According to the NCI, types of differentiated thyroid cancer, such as papillary and follicular thyroid cancer, are generally highly treatable and often curable, especially when the disease is found while it is still localized. Prostate cancer tells a similar story. When the tumor remains confined to the prostate, the long-term outlook is very good, and treatment can potentially be curative. But perhaps one of the most striking examples comes from rectal cancer with a deficiency in the DNA mismatch repair system, known as DMMR. In one trial involving 12 patients with stage 2 or 3 rectal cancers with this characteristic, all 12 achieved a complete clinical response after treatment with dostarlimab. And at the time of reporting, no disease recurrence had been observed during a follow-up period ranging from 6 to 25 months. These results give us an important clue. The problem is not that humans have no idea how to treat cancer. We have already learned how to treat some cancers extremely effectively. The problem is that there is no single treatment that can work for every type of cancer. We often talk about cancer as if it were one single disease. But in reality, cancer is a name for a very large group of diseases. Blood cancer is not the same as brain cancer. Lung cancer is not the same as rectal cancer. And even two people with the same type of cancer can have very different cancer cells. Imagine a tumor as a city. From the outside, you might think it is one single city, but inside are millions of residents with different characteristics. Some groups grow very quickly. Others grow more slowly. Some are sensitive to a drug, while others carry changes that make them much harder to destroy. Scientists call this phenomenon tumor heterogeneity, and this is why a drug can work extremely well in one patient, but be much less effective in another, even when both patients have been diagnosed with the same type of cancer. Now imagine that doctors find a drug capable of destroying most of the cancer cells. The tumor becomes smaller, test results improve, and everything seems to be moving in the right direction. But hidden inside the tumor may still be a very small group of surviving cells. These cells may already be less sensitive to the drug, or they may change during treatment. As the drug-sensitive cells disappear, the more resistant cells gain more space and resources to grow. After some time, the tumor may return. This is one of the important mechanisms behind treatment resistance. And what makes this even more frightening is that cancer does not always remain static during this process. Every time a cancer cell divides, new changes can appear. Some make little difference, but others may help the cell survive better in a harsh environment or resist treatment. When a drug eliminates the sensitive cells, the process creates a form of selective pressure. The cells that are easier to kill disappear first, while the cells that can withstand the treatment have a greater chance of surviving and continuing to divide. In other words, we are not simply trying to destroy a tumor, we are trying to defeat a population of cells that can change. At this point, a very reasonable question appears. If the drug is not strong enough, why not simply use a stronger drug? The problem is that cancer exists inside our own bodies. A treatment powerful enough to destroy cancer cells can also damage healthy cells. So the challenge is not simply to find something strong enough to kill cancer. We have to find a way to destroy cancer cells while minimizing damage to the body we are trying to save. This is also why cancer treatment must always balance effectiveness against toxicity. But even if we solve that problem, there is still another challenge because cancer cells do not fight alone. Surrounding cancer cells are blood vessels, immune cells, connective tissue, and many other types of cells. Together, they form what is known as the tumor microenvironment. This environment can influence how a tumor grows and how it responds to treatment. And that creates another paradox. The immune system is designed to detect and eliminate abnormal things inside the body, but cancer begins from the body's own cells. It is not completely foreign. Some cancer cells can evade recognition by the immune system or create an environment that makes immune cells less effective. This is one reason immunotherapy has become one of the most important directions in cancer research. But even immunotherapy does not work the same way in every patient. One person may have a powerful response while another may barely respond at all, and in some cases the disease can begin progressing again. That is why results such as the dostarlimab trial are so important, not because they prove that cancer has been cured, but because they show that when we identify the right biological characteristics of a tumor, a treatment can produce extremely powerful results in the right group of patients. But even when we control the original tumor, there is still a bigger problem. Metastasis. Imagine discovering a fire in one room. You can put out the fire in that room, but if sparks have already traveled to other places, the problem is no longer limited to a single location. Cancer can behave in a similar way. Some cancer cells can leave the original tumor, enter the bloodstream or lymphatic system, travel to another organ, and form a new tumor. This is called metastasis. What makes this especially frightening is that these new tumors do not necessarily look or behave exactly like the original tumor. They exist in different environments, face different pressures, and may respond differently to the same treatment. Now the question is no longer simply, how do we destroy this tumor? It becomes, how do we find and destroy every cancer cell that may already exist somewhere else in the body? And this is why the direction of cancer research is changing. Instead of viewing a tumor as one single mass of cells, scientists are increasingly trying to analyze it as a population made up of different groups of cells. Single-cell sequencing technologies can help researchers understand the changes occurring inside individual cells, while spatial mapping techniques can help determine where those cells are located and how they interact with their surrounding environment. The goal is no longer simply to ask, what drug can kill cancer? Instead, we have to ask, what types of cells exist inside this tumor? Which cells are resistant to treatment? How are they changing? And how can we stop them from adapting before they come back? This may be the future of cancer treatment, not a single superdrug capable of curing every type of cancer, but increasingly precise treatments designed around the characteristics of each patient and each tumor. So why can't we cure cancer yet? Because this question has actually been answered in part. We have cured or very effectively controlled some types of cancer. We have turned many others into diseases that can be managed for long periods of time. And we continue to develop new treatments. But cancer is not a single enemy. Every type of cancer has its own characteristics. Every tumor contains different groups of cells. And even within the same tumor, surviving cells can continue to change. That is why the fight against cancer may not be a search for one universal cure. Instead, it may be a search for a precise enough way to defeat each type of cancer, each tumor, and even each different population of cells within it. Not because we have failed to find a drug powerful enough, but because we are trying to solve a problem with far too many different versions. If you want to explore more scientific questions that seem simple but have far more complicated answers than we expect, subscribe to the channel and join us for the next story.